Welcome to Silent Vibes. In this video tutorial, I am going to discuss about a very most important enzyme used in the recombinant DNA technology, that is the restriction enzyme. So let's see, what is restriction enzyme? What are the functions of restriction enzyme? A restriction enzyme is also known as restriction endonuclease or restrictase. And the term restriction endonuclease is what? And why we name it as restriction endonuclease? As it is a nuclease, and in nuclease it has site, generally double-stranded DNA. Hence it only targets in double-stranded DNA. It is DNase as it acts only on the DNA as a template. And the restriction endonuclease, here endo means it cuts inside the DNA and cut both the strands. And why we call it as restriction? As restriction means basically it is a prokaryotic enzyme. It is only present in prokaryote, as prokaryote produces inside the cell. The restriction enzyme is an enzyme that cleaves DNA into fragments at or near specific recognition sites within the molecules. The restriction enzyme are one class of the broader endonuclease group of enzymes, and these enzymes are called restriction enzyme because they restrict infection of bacteria by certain viruses, that is bacteriophages. And how? It is by degrading the viral DNA without affecting the bacterial DNA. As I told you, it is a prokaryotic enzyme and it only present in prokaryote, and more than 2000 different restriction enzymes were discovered. And the discovery of restriction enzyme is done by 3 scientists. The first one is Arbor, the second scientist is Smith, and the third one is Nathans. And the restriction enzyme was first time identified by Arbor, and Smith isolated restriction enzyme, and Nathans describes the applications of restriction enzymes. Then the restriction enzymes cut the double-stranded DNA at a specific locations, and these enzymes predictably cut both the strands because the sequence they recognize is palindromic. And inside a prokaryote, the restriction enzymes selectively cut up foreign DNA in a process called as restriction digestion, and together this process form the restriction modification system. And palindromic is what? We will be discuss later about the palindromic sequence, so stay tuned till the end of the video. And how the entry of phages into the E. coli? Here the retrovirus enters by the two modification system, that is DAM or DCM, into the E. coli via the transduction method, where they restrict the infection of bacteria by certain viruses, that is by degrading the viral DNA without affecting bacterial DNA. And together this process form the restriction modification system. The first one is DCM methyl transferases at the cytosine at 5th position of the methyl group, and the second one is DAM methyl transferase in the adenine at 6th position of the methyl group. In the restriction modification system, the first is restriction of phage DNA, and here the phage injects the DNA into the bacterium, where restriction endonuclease bind to the phage DNA, and the phage DNA is cleaved and then it is inactivated. And the second, if the bacterial DNA is not cleaved, then the restriction endonuclease cannot bind to the recognition sequence, and the recognition sequence are not methylated. Then how we do the naming of restriction enzyme? What is its nomenclature? Here each enzyme is named after the bacterium from which it was isolated using a naming system based on bacterial genus, species and strain. And since their discovery in the 1970s, many restriction enzymes have been identified. And the first term, that is in caps E, comes from the Escherichia, that is a genus. The second term CO stands for coli, it is a species. And the third term capital R, it stands for RY13, it is a strain. And the fourth is 1. Here it shows the first identified as the order of identification in the bacterium. As we all know that there are two types of strain, rough and smooth, and here capital RY13, Here it is a rough strain. And by visualizing this, we can identify any restriction enzymes, that what is its genus, its species and its strain, and from which strain this microorganism belongs to. And so in this way, the naming of the restriction endonuclease does. As the restriction endonuclease are site specific, it is a sequence specific. It needs particular sites to cut. The restriction enzyme recognizes a specific sequence of nucleotides. It produces a double-stranded cut in the DNA, and the number of bases in the sequence will determine how often the site will appear by chance in any given genome. And the recognition sequence can be identified by the number of bases in the recognition site, usually between 4 and 8 bases. And restriction sites, for example, a 4 base pair sequence would theoretically occur once every 256 base pair, and if it is 6 bases, then it occurs in every 4096 base pairs, and if it is in 8 bases, then it would occur once every 65536 base pair, and the restriction sites are modified or unmodified. So these are the restriction sites. Mostly it is of 4, 6 or 8 base pair, and it is a palindromic sequence. So it is what? Suppose a DNA strand having 5' and 3' end, and if we move from 5' to 3' end in any sequence, then the sequence are same, such as TTAGCAC, which is same on both the strands. And as the palindromic sequence is a nucleic acid sequence in a double-stranded DNA or RNA molecule, whereby reading in a certain direction on one strand is identical to the sequence in the same direction on the complementary strand, the restriction sites are modified. And where it is modified? It is modified in the genome of bacteria and prokaryotes. We have earlier discussed about the modification, which is of two types, that is DAM and DCM methylation. In DAM, it methylates at adenine of the 6th position, and in DCM methylation, it methylates at the cytosine of the 5th position of the methyl group. The restriction enzymes are cutting within the site or outside the site. The restriction enzymes does not cut modified sequence or does not recognize as well. Because of this, the modification is done on the prokaryotic genome. Then what is the nature of cut? How the restriction enzyme cut? It can cut either in blunt ends or in sticky ends. In sticky ends, they have no overlaps that allows the two ends to base pairs and join together with another DNA strand. And in a sticky end, it is also known as the staggered or the cohesive ends. Here it is cutting both the strands at different position. It gives the overhanging results. It is overhanging from 5' end called as 5' overhang, and if it is from 3' end, it is called as 3' overhang. And the blunt ends are universally compatible with other blunt-ended DNA, and it possesses a 5' phosphate group to promote the ligation. The blunt ends, here blunt ends generate means blunt, that is parallel, that is both the strands are parallel. It cuts the two strands at same position, both in upside and downside. And the sticky ends get their name because they have overlaps, and the blunt ends have overlap which is universally compatible with other blunt-ended DNA. And in the sticky ends, the restriction enzymes such as BAMH1, BG3, CLAV1, EcoR1, HIND3, PST1, and these are the restriction sites. In the blunt ends, the restriction enzymes such as DRA1, EcoR5, HAE3, PVU2, SMA1, STU, and these are the restriction sites which are as follows. And how the production of blunt ends, and how the sticky ends produced, how the same sticky ends produced by different restriction enzymes. By using the restriction enzymes such as EcoR5, EcoR1, ALU1, we can produce the blunt ends or the sticky ends by cutting at a particular sites. Then how do we classify the restriction enzymes? Naturally occurring restriction endonucleases are categorized into 4 groups, that is type 1, type 2, type 3 and type 4, which is based on their composition and enzyme cofactor requirements, the nature of their target sequence and the position of their DNA cleavage, which is relative to the target sequence. And all the enzymes recognize specific short DNA sequence, carry out the endonucleolytic cleavage of DNA to give specific fragments with terminal 5' phosphates, and they differ in their recognition sequence, subunit composition, cleavage position and cofactor requirements. Then about type 1 restriction enzyme. And the type 1 restriction enzyme, its abundance is less common than the type 2 and its recognition sites, as it cuts both the strands at a non-specific location, that is either in 1000 base pair sites, as the type 1 restriction enzymes were the first to be identified and were first identified in 2 different strains, K12 and B of Escherichia coli, and these enzymes cut at a site that differs and is random distance, that is at least 1000 base pairs away from their recognition site, and these enzymes are multifunctional and are capable of both restriction, digestion and modification activities, that is depending upon the methylation status of the target DNA. Its cofactor is ATP, magnesium ions and SAM, and its nuclease subunit structure, as it is a heterotrimer, and yes, it has a DNA translocation, and its site of methylation is at the recognition sites. In the type 2 restriction enzyme, as it is most common, as its abundance is most common, and its recognition site, as it cut both the strands at a specific location, and it separate nuclease and methylase as its restriction and modification site. Its cofactor is magnesium ions. Its nuclease subunit structure, as it is a homodimer. It does not have the DNA translocation. Its site of methylation is at its recognition site. And typically type 2 restriction enzyme differs from the type 1 restriction enzyme in several ways. Type 2 restriction enzymes are very diverse in terms of amino acid sequence, size, domain, organization, cofactor requirements and the mode of action. And these enzymes cleave the phosphodiester bonds of double helix DNA. And these are the examples of type 2 restriction enzyme such as ALU1, HAE3, BAMH1, HIND3, EcoR1. And in the type 3 restriction enzymes, as its abundance is rare and its recognition site is cleavage of one strand, and the restriction and modification as its separate enzymes sharing a common subunit. In the type 3 restriction enzymes recognize two separate non-palindromic sequences that are inversely oriented. They cut the DNA about 20 to 30 base pair after the recognition site. The type 3 restriction enzyme recognize two separate non-palindromic sequences that are inversely oriented, and its cofactors are magnesium ions, and it is a heterodimer, and it does not have the DNA translocation. Its site of methylation is at its recognition site. The restriction sites can be further categorized into recognition site and the cutting site. For example, such as isocysomer, neocysomer, isochordomer, and the star activity. In the isocysomer, what is it? Isocyzomers are the pairs of restriction enzymes which is specific to the same recognition sequence. Isocyzomers are isolated from different strains of bacteria and therefore may require different reaction conditions. In some cases, only one out of a pair of isocysomer are recognized, both the methylated as well as unmethylated form of restriction sites. For example, SPH1 and BBUL1 are isocyzomers of each other. And what about neocyzomers? Neocyzomers are the restriction enzymes which recognize the same site and have a different cleavage pattern. For example, SMA1 and XMA1 are the neocyzomers of each other. Then the term isochordomer. Isocodamers are the pairs of restriction enzymes that have slightly different recognition sequence, but upon the cleavage of DNA, it generates identical overhanging termini sequences, and this sequence can be ligated to one another, but then form an asymmetrical sequence that cannot be cleaved by a recognition sequence. This sequence can be ligated to one another, but then form an asymmetrical sequence that cannot be cleaved by a restriction enzyme. For example, SAU3A and BAMH1 are the isocodamers of each other. Then what about star activity? The term star activity was introduced by Mayer, who characterized the modified activity of EcoR1. Star activity is the relaxation or alteration of the specificity of restriction enzyme mediated cleavage of DNA. The star activity can happen because of the presence of magnesium ions. Then what are the applications of restriction enzymes? As the restriction enzymes are utilized for gene insertion into the plasmids during the cloning and protein experiments, they are also used for the SNPs analysis, that is the short nucleotide polymorphism analysis, and identifying gene alleles. And the restriction enzymes are also used for the restriction fragment length polymorphism RFLP analysis. Thank you for watching this video. If you like the video, please do like, share and subscribe our channel. And further vital topics of the recombinant DNA technology as highlighting CSIR syllabus will be coming, so stay tuned for the upcoming videos.